Hello, I'm Dr. Philip Blair. I'm going to present to you about beta-Caryophyllene in this lecture. We'll get started now. So the title of my talk is Appetite and Metabolic Modulation with beta-Caryophyllene. I've been working with this product for about 3 years, and I've had extraordinary benefits and seen some incredible findings as a result of the treatment with beta-Caryophyllene in a broad number of cases. Hello, I'm Colonel Blair, and I want to talk to you about caryophyllene and its benefits to the body, particularly on appetite and metabolism. So let's get started. So the topic is appetite and metabolic modulation with beta-Caryophyllene. I want to go into this in detail, and I'm going to tell you all about the mechanisms that are at play in this process. First of all, my background. I graduated from West Point in 1972. I finished medical school in 1978. I was active in the Army as a combat physician, and I retired in 1996. I joined Elixinol with CBD in 2014, and I developed my own product, BC Plus, which is a formulation of caryophyllene in 2021, and I've been using it consistently ever since. I wrote a book in 2020, Medicinal Cannabis and CBD and Mental Health Care with Kylie O'Brien, and this is available at Amazon or Springer areas of books, and I highly recommend it. I do mention beta-Caryophyllene, but the focus is on CBD and mental health care. So Celine, this woman that I've got pictured here is Celine Green, and she's a 36-year-old mother, entrepreneur. She lost 15 pounds over 4 weeks, and she controlled her ADHD and her nicotine habits using beta-Caryophyllene alone. And that was the only thing she was doing. And the spectacular sort of benefits in terms of controlling her habits, as well as her appetite, and the overall benefits to her well-being and her performance were phenomenal. I've also had a great deal of other successes, and I list those off where KJ had a lost weight of 12 pounds. Another woman lost 13 plus pounds, and another woman lost 45 pounds over 3-year time period. So the benefits are profound, and it has lots of effects as benefits as well in terms of relaxation, calmness, and mental focus, as well as the weight loss performance. Most of these people were not actually trying to lose weight, and that's what I bring to the table is an active metabolizer for the body. First of all, what is caryophyllene? Well, it's an essential oil. It's common in many spices. It alleviates pain, inflammation, anxiety, and metabolic disorders particularly. It's found in thousands of plants, but it's got high concentration at about 36 species, particularly cloves, hops, even avocado, and hemp. There are over 3,200 articles that have been published on the topic, and they're all beneficial, and they all have performance aspects. Unfortunately, there are only 10 that have been human studies, and so we're really deficient on human studies in this product. It's FDA approved as a food flavoring, but it has no toxicity. It's available to the lungs, to the skin. It hasn't produced any reproductive ill effects, and it's anti-mutagenic, so it's going to prevent and repair DNA damage. It's also it joins with a chemotherapy for intracellular concentrating effects, but it protects against toxicity. And it does have a CYP3A4 inhibition, but that has not interfered with any drug metabolism, and there's been no drug reactions that I've found with it. So how does beta-Caryophyllene work? Well, it's pleomorphic in terms of its targets. It's got over 100 targets, and I'm going to be listing off a number of additional targets. Now, these targets are cytokines, enzymes, genes, signaling molecules, adhesion molecules, chemokines, and signaling pathways. And I list those off, and particular receptors. Now, it is a CB2 specific and very strong cannabinoid type 2 receptor agonist. And so that's the key point that I offer you now. What are the mechanisms of appetite? Well, there's the physiologic mechanism, which means that we're going to have a balance of energy determination, and we're going to have appetite that's controlled by that. So when we're depleted of food for a long period of time, we're going to get hungry and we're going to want to eat. When we eat, we're going to be satisfied. That's the homeostatic mechanism, but so often that we are involved with other processes in the appetite control. And that mainly belongs to the reward pathway. And this is commonly called the addictive pathway. And third category is inflammatory. Beta-Caryophyllene addresses all three pathways succinctly and effectively. Now, these are some of the hormones that are involved with the pathways, the physiologic pathways with appetite. Ghrelin, leptin, GLP-1, peptide, tyrosine, tyrosine, and neuropeptide Y intracellular in neurons in the hypothalamus. And the melanocortin pathway is also a stimulus for appetite, and adiponectin. So when we eat something, well, before we eat something, if our stomach is empty, we're going to release ghrelin, and that's going to stimulate our appetite. But when the food gets into the intestines, we're going to release these specific hormones to temper our appetite and control and seek satisfaction. And the primary one is GLP-1. So GLP-1 controls much of our appetite, and its activation actually stimulates satiety, and it decreases the appetite, but it also provides a number of other mechanisms that provide neuroprotection for the body. And those neuroprotections are being manifest in drugs like Ozempic, which have been promoting a great deal of weight loss as well as benefits to the individuals. The problem with Ozempic is that it actually reduces gastric emptying. It's a gastric acid secretion, and there's gastric motility that's impaired. So that can lead to nausea and vomiting and diarrhea. And more recently, there have been some studies that have showed that Ozempic has had problems with depression, suicide ideations, and the weight loss that is associated with it is actually muscle loss as well as fat loss. So it's a derogatory type of fat loss. And when you stop the GLP-1, you regain your weight entirely. So the reward pathway deals with the brain, and it starts at the brainstem at the ventral tegmental area, and that stimulates the nucleus accumbens, and that is also controlled by the prefrontal cortex. And the prefrontal cortex is important for our inhibition of our impulses. It's weighing our consequences. It's reflective decision making. But so often this is impaired, or this particular process is paralyzed or inhibited in the appetite, in the reward pathway, so that it's not functioning properly in the body. Now, another pathway is inflammation, and inflammation causes the toll-like receptor 4 to be activated, and this induces an inflammatory signal that actually increases some appetite suppressants, and it can decrease the appetite. But this is highly variable in many people, and there is considerable evidence that it can have modified effects on the body. in particular, the inflammatory pathway, if we get an infection or we get an injection with some toxin, we're going to decrease our appetite. If you're very, very sick and in the ICU, you don't have much of an appetite. And that's when we actually have to stimulate the appetite. We have to get food in, and we may have to deliver intravenous food supplements. But more often, we're dealing with a high-fat diet, and we're dealing with obesity, which is also an inflammatory pathway that actually increases the appetite and decreases the energy expenditure. So it's something in between there, but it's important to recognize that the toll-like receptor 4 is that primary agent and a primary factor that controls the inflammatory signals leading to this appetite increase. Now, what are the effects of sucrose, glucose, and fructose on the peripheral and central appetite signals? Well, rats had an elevated serum levels of fatty acids and triglycerides and cholesterol. And in spite of that, they had increased in ghrelin, so they're increasing appetite, increasing leptin, and they've had decreased the PYY, which is going to be controlling for appetite. It downregulates the hypothalamic NPY and POMC receptors at the mRNA, and that's going to also decrease the appetite suppression. So you're going to have increased appetite, and you're going to actually upregulate the CB1 receptors in the body. And we know that CB1 receptors actually stimulate our munchies, and they have a number of adverse effects, particularly on the liver and on the gastrointestinal tract. And so by stimulating these receptors, we're getting into a negative sort of situation for the body weight and modulating the body weight. So how does beta-Caryophyllene work? It works on appetite regulation. I'm going to go at this from three angles, from the physiologic, the reward system, and the inflammatory system. Well, beta-Caryophyllene actually regulates GLP-1, and I've listed them off here, the CB2 receptors. And these particular receptors, it controls and regulates these particulars. I mean, all of them. And so it's regulating all factors that have to do with appetite. So first off, it upregulates GLP-1 and adiponectin. It upregulates the melanocortin pathway that also reduces food intake. And it downregulates the inflammatory receptors. It downregulates leptin, and it shifts the macrophage from M1 to M2. That's the anti-inflammatory phase in adipose tissue. So it's reducing the inflammation from adipose tissue, and it's reducing the inflammatory signals that come from it. The CB2 receptor is also extremely important because it is going to inhibit the reward pathway. Now, there's good documentation that it prevents opioid, cocaine, nicotine, methamphetamine, alcohol, benzodiazepine, and even THC addiction. And there's even literature that supports it as a food, inhibiting food addictions, particularly to fructose and other carbohydrates. But the strength of this is very, very strong. And as a CB2 receptor, blocking these particular reward pathways is incredible and has great potential for decreasing appetite, decreasing the sensitivity of these foods and these substances to the body. Now, I really like to get after the CB2 receptor because I think it's a major receptor that provides all the benefits of the body, but it's not well recognized in the brain. In fact, the CB2 receptor is located throughout the brain, and oftentimes it's devoted, and many of the medical articles that I read say that it's just in the periphery. No, it's quite populous in the brain. It's all over. It's in neurons, it's in synapses, it's in glial cells, and it's even in the vascularity of the blood-brain barrier and the endothelial cells. So it's a very, very key and important activator for the benevolence of this particular substance, of different activators of the product. So it's going to control for many, many adverse effects. Now, in neurodegenerative diseases like Alzheimer's and Parkinson's, this receptor system becomes very, very highly populated. And so it looks like it's an opportunity. If we could connect with the CB2 receptor activated, then we're going to reduce our inflammatory and our neurodegenerative type of problems as well. So the role of CB2 cannabinoid receptor in the development of food addiction in male mice, this was studied in depth, and it showed that imbalances in the brain reward pathway are commonly shared across food and drug addiction. And CB2 overexpression decreased the CB1 receptors and the opioid receptors in the ventral tegmental area of the body. And it also it creates a potential target for eating control and comorbid emotional effects associated with food addiction. So like cravings and abnormal behaviors and depression, all these things can be controlled and regulated with CB2 activation. So the abuse of substance, CB2 controls the abuse of substance, foods triggering reward effects, through the dopamine behaviors. And the CB2 and the dopamine receptors are co-located in so many different key parts of the brain, particularly in the ventral tegmental area and the NCA process, which are the key areas for addiction. CB2 also regulates the impulsive seeking by modulating of the dopamine system as well. So that reward system, that pleasure system, is controlled and regulated by the CB2 receptor. And in addition, the CB2 receptor is going to be active at the ventral tegmental area, and the dopamine neurons are going to project to the nucleus accumbens, and it modulates the DA tone. So it's modulating this tone to prevent for that super high that you get from eating something pleasurable. So it's going to control this factor, and it's so superb in controlling addictions of all kinds. So I want to move on to the the toll-like receptor 4, because that's the inflammatory pathway. And beta-Caryophyllene there's the evidence for it here, where the beta-Caryophyllene actually ameliorates the inflammatory signaling by the TLR4 and the nucleic factor kappa beta, as well as the inflammasome, the NLRP3 signaling pathways. And it's protecting against stroke, actually, by polarization of the microglia towards the M2 phenotype via the TLR4 pathway. And this is I just want to give this support and this evidence for its major role in the anti-inflammatory effects. So we talked about appetite. Now let's get into metabolism. And there's so many different mechanisms of metabolism control with Caryophyllene. I just list off a few of those particular pathways, but I want to get into some depth into them. So first off, the GLP-1 is a major regulator of the metabolism of the body. Not in addition to decreasing the food intake, it's going to increase skeletal muscle glucose uptake. It's going to reduce glucose production from the liver, whereas it's going to increase insulin secretion, and it's going to decrease glucagon secretion. It's also going to convert white fat to brown fat with a thermogenesis. So it's going to create an energy situation that's going to burn up calories and energy quite rapidly. So within diabetics, we know that the GLUT4 is the receptor for the glucose molecules, and these are upregulated in muscle cells in diabetics. In addition, in diabetes, it's preventing oxidation and stress and the inflammation in pancreatic beta cells. So it's going to preserve the insulin secretion, but it's also going to activate the insulin sensitivity for the body. So it's going to improve all aspects of diabetes as well. Now, I've talked about the upregulation of GLP-1, but also about adiponectin. Adiponectin is a marvelous molecule that is highly effective in terms of coupling with metabolic factors as well as appetite. So high levels of adiponectin decrease the appetite, but they also provide a number of other benefits for anti-inflammatory changes as well. Beta-Caryophyllene is going to increase the mitochondria, but it actually decreases the reactive oxygen species. It also upregulates the mitochondrial uncoupling protein. The uncoupling protein is that one that releases energy as heat rather than as being burned energy, and reactive oxygen species can be generated from that. It's going to actually increase energy expenditure, and that means weight loss. And the browning of fat, browning of white fat to brown fat is particularly noteworthy because this may be the key factor in why really weight loss occurs. I provide this slide in evidence that beta-Caryophyllene is an agent that is going to be activating adiponectin. It's going to increase the levels. And also the nuclear factor erythroid, the NRF2 signaling pathways is key and important to reduce oxidative stress. And it actually activation prevents a number of programmed deaths of cells. Now, in this particular study, beta-Caryophyllene actually activated the signaling pathway, but adiponectin increased the anti-inflammatory cytokines by 144-fold over the control animals. And in addition, and this is just an aside, beta-Caryophyllene inhibited anaphylactic-like shock induced by this chemical. And it also inhibited all mast cell degranulation and histamine release. So this is a side benefit of beta-Caryophyllene in blocking allergic phenomena and even anaphylactic shock. So what about dealing with diet? Well, these rats were fed a high-fat and fructose diet, and they developed behavioral alteration and insulin resistance. In each case, CB2 activation by beta-Caryophyllene reduced and inhibited those particular actions. In particular, that you see that it reduced oxidative stress, lipid peroxidation, and inflammation, and whereas it actually activated the PPAR gamma receptors, which are key and important for our energy determination and our mitochondrial function. It also activated brain-derived neurotrophic factor, which is what we generate when we exercise as well. So the PPAR alpha, this is a nuclear receptor that has great value on the body, and it produces a cascade of different events on the RNA and on the DNA, whereas it actually reduces oxidative stress by increasing antioxidant enzymes and reducing reactive oxygen species and lipid peroxidase. Now, on the other hand, it's actually increasing glucose metabolism, and it's actually increasing lipid metabolism in the form of fatty acid oxidation, as well as HMG-CoA synthesis and ketogenesis. So it's stimulating autophagy, which is a key process in the brain for developing new signals and clarity to the brain and avoiding degenerative changes on it. Now, it also affects PPAR gamma, and PPAR gamma is a key factor in so many different mechanisms and different processes, and it's probably the most influential factor at all. Now, CBD also connects and activates PPAR gamma, but beta-Caryophyllene does a superb job. On the left, you see the macrophage decrease in inflammation and activation of M2. And at the top, you see the muscle cells stimulating insulin sensitivity, glucose-stimulated insulin secretion, and then over on the adipose tissue, you get an increase in brown fat and lipid metabolism. And down at the bottom on the right, you see that it decreases actually food intake, and it decreases inflammation. So why do giant pandas roll in horse manure? Well, they roll in horse manure because it contains beta-Caryophyllene and beta-Caryophyllene oxide. It works as an antagonist for the TRP, TRP 8 M8 receptor, which activates it's in order to cope with low temperatures. And what they do by these when it gets to really low temperatures, they can actually generate a great deal more heat and stimulate the production of brown fat. Now, we do the same thing, and that's probably what the benefits of cold immersion in the stimulation of brown fat, because brown fat stimulates systemic metabolism. It increases the energy balance, it decreases body fat, it actually is anti-diabetic, and it stimulates thermogenesis, so that excess burning of calories that are in the body. This is probably one of the major factors for weight loss with respect to beta-caryophyllene and cold exposures. So if you're talking about the TRPM8 receptor system, then it's going to be sensitizing the body, desensitizing the body to cold temperatures, but it's also going to be stimulating cell proliferation with vascularization, mitochondrial content, oxidative capacity, lipolysis, lipogenesis, and glucose uptake. So all the things that you want to do in an athlete or in somebody who is overweight and having problems controlling their weight. So these are very similar to ketone effects. And what I've seen with beta-caryophyllene is it's a fasting mimetic. So it actually simulates what goes on in fasting. And the parallels are of ketone bodies, beta-hydroxybutyrate and beta-caryophyllene are very, very similar. And I've put these listed on this page here where I've got 13 particular mechanisms that are being activated by beta-caryophyllene and beta-hydroxybutyrate. So they're extraordinarily coupled, as well as, so there seems to be some crossover, but we don't have any evidence for beta-caryophyllene actually stimulating beta-hydroxybutyrate. But there's no doubt that there's something going on here. But I wanted to parallel the particular benefits of ketone bodies and beta-caryophyllene. So I've gone over quite a bit, and I just want to summarize here with the beta-caryophyllene targets in the metabolism. It's going to target adipose tissue, shifting from white fat to brown fat. It's going to activate the melanocortin system, which has a lot to do with our perceptions of appetite and also our metabolic functions. It's going to reduce reactive oxygen species and leptins. It's going to reduce cytokines, so that an inflammatory network, and it's going to increase adiponectin receptors. And so you're going to get that benevolence of the control of inflammatory signals as well as appetite signal suppression as well. And it's going to particularly target the uncoupling proteins in the mitochondria. It's going to stimulate the mitochondria and cause proliferation of the mitochondria, and it's going to act on the nuclear receptors, the PPAR nuclear receptors, and the NRF2 receptors in all cells and in the brain. So the summary is that it has extraordinary particular properties with regard to weight loss. It's going to decrease food cravings. It's going to improve energy. It's going to decrease body fat, and it's going to increase fat breakdown. It's increasing the number of mitochondria. It's reducing dysfunction, and it's going to modulate our hormones, the adiponectin, the leptin, and the GLP-1. It's an incredible single substance that has adaptogenic properties that are extraordinary. So that's just on appetite and metabolism. I've done a great deal of research and found that it actually beta-caryophyllene is effective in all of these problems as well, and highly effective in them. But there is not any medical human studies on this particular product at the present time. What we need to do is get these human studies done to prove that they have these benefits. But with so diverse a topic from a natural food substance, it should be a no-brainer that this should be used. So it is a natural herbal food substance. It modulates the neurometabolic and immunologic system. It's FDA accepted. It's got low dosing, few adverse effects, and it's an alternative to pharmaceuticals, incredible alternative to pharmaceuticals. It is established in its effective liposomal format, so it's got a really good formulation that could be highly, highly effective. So I offer 4 products. I offer in crunches, which are small edible cookies. I offer the pure Caryophyllene tincture, which is the essential oil. I offer it in a gel as for topical application, and I offer the oral liposomal blend, which is the primary product that I'm discussing here. The whole idea of this particular company that I'm involved with is to create balance and complement health through working through the endocannabinoid system. Now, BCP works really well with CBD, and it can be highly effective in combination. So what I'm trying to do, and I hope you share in those thoughts with at the Cannabis Public School, is that if I can promote it in some way, I would love to have you ask questions, and we can inform you on further developments in this product development. Thank you very much.